मैं लगा दूंगी. Good morning, everyone. I hope you all had a hearty breakfast and are energized for another exciting day of learning. I'm Neha Ghosh, your host, and I warmly welcome you all to the second day of our first genomic workshop on NGS and bioinformatics. Today, we'll delve deeper into the technical aspects of next generation sequencing, focusing on the essential processes and best practices crucial for successful genomic analysis. We'll begin with a session led by Mrs. Manisha More, who will guide us through the critical steps of library quantification and quality assessment, along with strategies for effective interpretation of results.
A warm good morning to all of you. Myself Manisha More. Today I will be talking on quantification and quality assessments of library. The overall workflow of NGS starts with an experiment starts with the sample preparation, that is isolation of your nucleic acid, library preparation, sequencing, data analysis and interpretation. Assessing the quality of the library at this stage is crucial because it determines the success of the subsequent sequencing run. So what is library? Library represents the DNA or the RNA fragments that are ready for sequencing. So basically, library corresponds to a single sample and a multiple libraries, each with their own unique adapter sequences, and can be pulled and sequenced in the same sequencing run. These are different types of libraries which has already been discussed by Nikhil sir, like whole genome sequencing library, RNA-seq library, exome genome sequencing library. All depends upon your sample material and the panel which you are interested. What is library quantification? So library quantification is a process of measuring the concentration of DNA fragments to ensure optimal sequencing results. It ensures that the correct amount of fragment is loaded into the sequencer for optimal results. And why library quantification is necessary? Accurate library concentration, it determines the direct impact on the sequencing performance. Precise quantification of library is a critical step for achieving uniform sample pooling and optimal cluster density when sequenced. If the library concentration is too high, it may result in overloading the sequencing platform, leading to poor quality data. On the other hand, if the library is too diluted, it may generate enough data, resulting in a wastage of your sequencing run. So, how is library quantification carried out? There are different methods for quantification and quality control, depends on the sequencing library kits being used. Here are several different techniques by which quantification can be carried out, like ultraviolet spectrophotometry, fluorometry, electrophoresis system by quantitative PCR. Some of these, such as ultraviolet spectrophotometry, requires no modification of the samples, while others, such as fluorometry, requires a staining or dyeing of the sample with a molecular markers to enable more detailed inspection. The best choice of the given application depends on the many factors, including the degree of accuracy, capital expenditure on your equipment, cost consumables, and each of these has his own advantage and his specific cons. The first method is ultraviolet spectrophotometry, that is nanodrop. Nanodrop uses the principle of ultraviolet spectrophotometry. Nanodrop determines your both concentration and purity of the nucleic acid. The purity generally indicated in 2 ratio, that is absorbance at 260 by 280 for a nucleic acid, and for protein, the absorbance at 260 by 230. If the more light absorbs, more is the concentration. Library quantification using spectrophotometry based methods are subject to overestimate your library concentration and should be avoided. It is not accurate and not recommended for NGS library quantification. A ratio of around 1.8 is generally considered to be form of a pure DNA, and a ratio of around 2.0 is generally considered to be a pure form of RNA. If the ratio is less than 1.8, it is because due to your protein contamination. So proteinase treatment has to be given to your sample, and if the ratio is more than 2.0, then it might be a RNA contamination or phenol or residual of a phenol or other reagents used in your extraction protocol. So advantage of this, it is a quick, easy to use method, does not require expensive reagents, provide an estimation of DNA purity by calculating 260 by 280 ratio of your nucleic acid. Disadvantage, it measures total nucleic acid content including a single stranded DNA, RNA and contaminants, not as accurate for low concentration samples and for determine the amount of functional adapter ligated fragments. The second method is fluorescence spectrophotometry like qubit. So here the fluorescent dyes such as cyber green or pico green fluorescent dye can be used. So fluorescent dyes bind specifically to the double strand DNA in the sample, and the fluorescence emit is measured. The fluorescence intensity is directly proportional to the concentration of the DNA in the sample. Fluorometry methods are the best choice of for libraries with broad fragment size distribution. So qubit offers a specific quantification kit, depends on the type of biomolecule being measured. So for library quantification, double stranded DNA high sensitivity assay kit is recommended for your library quantification, and for concentration of your template, a broad range quantification kit is required. So fluorescence spectrometry, it is highly selective for double stranded DNA. DNA quantification is carried out using a fluorometer, which compares the fluorescence of a given sample to a standard curve and a set of a DNA sample of a known concentration. Here a use of positive control is recommended, such as a previously sequenced library or a commercially genomic DNA control can be used. Quantification limit is 0.2 to 200 nanogram. Advantage, it is quick and simple process, highly specific for double stranded DNA, reducing the interference from single strand of DNA or RNA. Minimal DNA sample consumption, only 2 microliter is required. Disadvantage, not sequence specific, cannot differentiate between adapter dimer, primer dimer and target libraries. The other method is electrophoresis system such as a bioanalyzer or tape station. So tape station is the advanced version of a bioanalyzer. So it is a microfluidic based automatic electrophoresis system. It is usually recommended to check the quality of this of your library, that is checking the library size distribution. The tape station system integrates an instruments, a data processing software, reagents and a screening tape that is specific for your DNA and RNA test. It is suitable to analyze size, quantity and integrity of your sample. High sensitivity DNA kit is recommended. So this is the overview of a screen tape, that is a front view and a back view, which is having a barcode which will give you a detailed information of the traceability of your results. Then it is a pre-filled, it is having a pre-filled reagents like buffer, electrodes and a gel. So no need of sample preparation, it is already pre-filled cartridge. So screen tape technology enables a less hand-on activities during the sample preparation. The device has a 16 lanes so that each sample is analyzed in an individual lane, completely eliminating contamination and carryover. The unique benefit of the tape station system is the flexibility to run any number of sample at a cost per sample. And if you are running 2 samples and the remaining 14 tape is blank, you can store it in a fridge and use it in the next time. So these are the screen tapes which are available depending upon the types of nucleic acid you are analyzing. So basically double stranded DNA kit is used, this 1000 kit is recommended. So this is the interpretation of your tape station results. A graphical plot shows that the fluorescent intensity of the nucleic acid The x-axis represents the fragment size in the base pair, and the y represents the fluorescent intensity, which correlate the amount of DNA or the RNA. And the gel image, it is a virtual image that's Sametris shows that how the fragment would appear in the gel electrophoresis system, and it shows the band corresponding to the different fragments sizes. This is an example of a good library. So here the summary is also provided with the quantitative information about the concentration of the samples, which is often expressed in nanogram per microliter. This is an example of a library in which there is an adapter dimer attached. So in this process, a cleanup procedure is required for this. This is a library which is not worked. Okay. So advantage of this is it provides a fast and convenient way to assess both the concentration and the quality of a DNA and RNA libraries. It is ready to use pre-filled separation matrix and running buffer electrolytes, no carryover due to its inbuilt liquid handler system. It has a 16 individual separation channels. Although 96 samples can be run in a single experiment, and it is a sample traceability due to its unique barcode system on the screen tape. So next quantification method is by ddPCR, that is droplet digital PCR. It contains all the necessary components to create a droplet and quantify your NGS library. So ddPCR is highly sensitive and precise method for quantifying NGS library. It is highly accurate measurements without the use of a standard curve, provides information about the library quality such as adapter dimers, and indicates the library insert size. It enables a precise balancing of your pool library. So this is a general workflow how a ddPCR works. So DNA templates is individually partitionized into a tiny droplets. Approximately 20,000 droplets has been created, where the target and the background DNA randomly distributes among the droplets. So after amplification of each partition, each droplets provides a positive and a negative fluorescence. Based on the ratio of the total droplets, the system calculates the absolute quality of the target molecule using a Poisson statistics. And this absolute concentration of the nucleic acid of the sample is calculated as copies per microliter and analysis by the Quantas of software. So this is the interpretation which consists of ddPCR data can be viewed as a 1 DD plot. That means there is only one target is amplified. The numbers are plotted on the x-axis, and the fluorescent intensity is plotted on the y-axis, and a red line is a threshold events. Above the red line are considered as a positive, and below, and the line one below the red line are negative. This is the second interpretation in which a ddPCR with two targets are amplified and can be viewed in 2 DD plots where 4 different clusters are generated. If at a time 2 targets are amplified, results can be viewed as 2D plots where 4 different clusters are generated. Signal from that is a FAM plot is on the x-axis, and signals from the hex plot is plotted on the y-axis. Advantage ddPCR is the endpoint measurements that enables use to quantify nucleic acid without the use of standard curves. The presence or the absence of digital format, no need of external calibrators and individuals, controlled, sensitive and accurate. It is sensitive and accurate. Disadvantage, it is a time consuming, multiple sample handling steps is required, and highly skilled person is required. Next method is quantification of library by qPCR with using a Kappa biosystem kit. The Kappa library quantification kit contains a library quantification standards. It has contains 6 standards which are serially diluted. With that, it contains a primer premix reagent and a qPCR master mix reagent. So the six pre-diluted DNA standards and approximately diluted NGS libraries are amplified using a platform specific qPCR primer that targets the adapter sequences. The average cycle threshold value of each DNA standard is plotted against its known concentration to generate a standard curve. And the standard curve is used to convert the average cycle threshold value for the diluted libraries to a concentration from which the working concentration of each library is calculated. Critical steps for success is accurate liquid handling is must. Libraries and control should be diluted to falls within the dynamic range of your standard. Library should always be diluted in a trace EDTA buffer to prevent degradation. Note NTC should be included in each run, and previously sequenced library should be included in every run as an internal control. Advantage is reliable and sensitive quantification of all sequence components library molecules. It is accurate and reproducible with equimolar pooling for multiplex sequencing. Disadvantage, it is time consuming, multiple sample handling steps is required, and it is 5 to 10 times more costlier than the spectrophotometric assay. To summarize is multiple methods are available for NGS quantification. Choice depends on the desired accuracy, cost, and application. Inadequate quantification can have a significant effect on your library generation, normalization, pooling, and relative coverage of depth for multiplexing, overall cost and limits for detection. Thank you.
Thank you, ma'am, for that informative presentation. Next, I welcome Mrs. Swapnali Joshi, who will walk us through the essential steps involved in loading sequencing ready libraries. Okay.
Nikhil sir, I just have one question. In this RQ method, the standards that we use are in-house prepared like plasmids, or what are the standards that are used?
If you use the kit that is there by the company, then you don't need to. Then you need to use those kits, those standards. If you do ddPCR, obviously there are no standards. The problem with that method is it's very expensive, because as we all know, you can't do real time in single plex. So you have to run replicates.
Sir, are you using this method?
Not at all. We use only qubit because it is very cost effective. We may use ddPCR. We are trying to develop something for ddPCR. We'll see how it goes, because ddPCR is very cost effective. So both address different things, qubit is only for quantitation. Tape station is only to assess the quality of the library. So if you want to see the spread of your library, whether it is proper or not, whether there are any adapter contamination, all for that you require tape station. Both address completely different things. Tape station we never use for quantitation. Qubit we never use for quality. It's only for quantity of DNA.
Like if I'm dealing with FFP samples, then
still the principle stays the same. At the end of it, you have to see a library. If it's an amplicon library, you will get one straight size. If it's a library that is even though it may be amplicon, if it has undergone fragmentation and repair retailing, you are going to see a spread of DNA fragments. But for that you need a library. If tape station is expensive, it costs 30-35 lakhs, and unfortunately complete monopoly of one company. So if you don't want to do that, Kaya Excel is there, but that is not really for libraries. It's not that sensitive. You can just do a page gel. So if you are comfortable doing page gels, you can get away with it just fine. You will get very good results.
We are using QSEP Plus, which was provided by Illumina only. It costed us around 12 lakhs, and we do library quality check on that. So it requires that special syringe for loading the samples. It is a little costly, but I think it can be a good alternative to tape station. It's not that costly.
Okay, how much does it cost you per sample?
Cost per sample, as in I have to calculate and tell you, but then we are routinely using it. If it was not cost effective, we wouldn't have been able to sustain it in long run. At least it's a good alternative to tape station.
Okay. That's good to know. QSEP.
QSEP plus, sir.
QSEP.
By Illumina only.
Okay.
So good morning everyone. Today I'll be talking about loading of sequencing ready libraries. So these are the few topics that will be covered in my presentation. First of all, basic workflow of the sequencing, pooling of the libraries, how to estimate the loading concentration of the library, how the libraries are loaded onto MySeq, MiniSeq and NextSeq, and what are the different versions of Illumina kits that are available in the market, and how SBS technology works. So Illumina NGS workflow includes 4 basic steps. First is library preparation, cluster generation, sequencing by synthesis, and data analysis. We have already seen the library preparation where the DNA is randomly fragmented, and to this fragmented DNA, platform-specific adapters are ligated, and these adapter-ligated fragments are then PCR amplified. During the cluster generation, which parts takes inside the instrument, the clonal clusters of individual DNA molecules are generated onto a flow cell using a pre-filled reagent cartridges on the sequencing platform. And in the sequencing by synthesis method, the sequencer reads the nucleotides one by one, and during the data analysis, the FASTQ that are generated after the sequencing run, these reads are filtered for quality, assembled and aligned to a reference genome. So library prep is the first and very critical step for successful sequencing. There are various standard and proprietary library preparation kits are available in the market for various NGS methods. But after the sequencing libraries are prepared, the actual sequencing stage remains fundamentally the same. So this is a typical structure of the Illumina library where you can see P5, P7 adapters which are platform specific. Then there are 2 index 1 and index 2 which are sample specific. These are the read 1 specific and read 2 specific primer binding sites, and this is the DNA insert which is DNA for interest. So for clustering of the libraries must have P5, P7 binding regions on the library which are complementary to the flow cell oligos. So during clustering phase, these libraries will go and bind to this P5, P7 adapters that are present on the library and flow cell oligos. For sequencing, libraries must have sequencing primer binding site, which are read 1 specific primer binding site and read 2 specific primer binding site. And for mixing of the libraries, library must have unique index or barcode sequence, due to which we can multiplex many samples together in a single run. And after using these indices, we can demultiplex them. So first of all, after getting a qubit count of any library, we need to convert that nanogram per microliter reading to nanomoles. So first of all, you need to quantify your libraries on high sensitivity DNA qubit assay and record the concentration of the nanogram per microliter for each library. Then we need to convert this nanogram per microliter library to nanomole before sequencing. So you can do it using this formula wherein you can see this is the concentration of the library in nanogram per microliter. Then 660 grams per mole is the average molecular weight of any DNA base pair. This is the average library size in base pair which you can determine using tape station. This will after this calculation, you will get your results in nanomoles. So using Excel sheet also you can do it, wherein I have written, this is the example. So for 3.24 nanogram per microliter of the library, you will get 17.53 nanomoles. So for most of the Illumina sequencing platform, 2 to 4 nanomole for each library is preferred starting concentration for denaturation and dilution of the libraries. So first of all, before pooling the library, you need to determine the number of samples that are sequenced together. Then you need to estimate the reads required per sample to achieve desirable coverage. Then total number of samples that are going to be pooled in single sequencer run. And you need to do the qubit quantification using high sensitivity assay. So this is the Illumina online calculator available. So that using that you can convert your nanogram per microliter libraries to nanomoles. So this is the example wherein library plexity stands for how many samples you are going to put in single pool. Then libraries will have different concentration. Unit of the measure of the library is like nanogram per microliter, which you can determine from qubit HS assay. This is the library size, which is determined using tape station, and we need to convert it to 1.25 nanomoles. So if you are going to put your libraries in MySeq, Illumina recommends converting your libraries to 1.25 nanomoles. So after that you will get your results in nanomoles. So before multiplexing the libraries, here are some things that you need to consider for Illumina sequencing. Illumina recommends pooling of the libraries that were prepared using same workflow. You need to use 1.5 ml DNA low bind tubes and carefully add required amount of the library. 1.5 ml low bind tubes are the specialized append of tubes that are designed to minimize the sample loss and maximize the recovery of the nucleic acids. Then also library should have the similar fragment size that should fall within Illumina specifications. The equivalent concentration has to be there for each pool to generate equal reads per sample. Then also ensure the final pool is free of contaminating adapter dimers and has an accurate size distribution, because this primer dimer can go and bind to your flow cell and it can create excess of the reads of the, and then it should have uniquely indexed adapters that are attached to your libraries. So now why accurate pipetting is must during the pooling. So in first panel you can see there are 3 samples, sample A, B, C, but those samples are not equimolar pooled in one pool. So because of which there is non-uniform data distribution. So sample B has got more reads than as compared to sample A and sample C. But in next panel you can see all 3 samples are pooled equimolarly, so there is equal distribution of the reads. So clustering efficiency also varies with the fragment length. Shorter fragments will cluster first on the flow cell as compared to longer fragments, and inaccurate pipetting will result in skewed results. So these are the 3 next generation sequencers that are available in the lab. This is MySeq, it uses SBS technology, it has 4-channel chemistry. This is MiniSeq, it also uses SBS technology but 2-channel chemistry. And this is NextSeq, using SBS technology and 2-channel chemistry. So now let's move on to how we load the libraries on the MySeq. So in the MySeq kit, it comes in the 2 parts. One is a reagent cartridge and one is flow cell. So reagent cartridge is nothing but the pre-filled sequencing and clustering reagent in one unit, and it also has a one tube of HT1 buffer, which is a hybridization buffer, and flow cell and PR2, that is incorporation buffer, is stored in 4 degrees refrigerator. For MySeq, flow cells are shipped in a buffer solution. So before loading onto a MySeq, we need to clean them thoroughly with the nucleus-free water. And MySeq reagent kit is single-use reagent kit, which is required to perform the sequencing run. And reagent cartridge is provided with the RFID identification for accurate consumable tracking and compatibility. So how to determine the optimal loading concentration? That is also a critical step to utilize reagents efficiently. Library loading concentration can be optimized for the MySeq according to the results you are seeing from your previous study or library type. So run parameters that are taken into consideration to determine optimal library loading concentration are cluster density, Q30 score, and cluster passing filter. So cluster density is a metric that measures the number of clusters per square millimeter on a Illumina flow cell. It is an important factor for determining the quality and the yield of your data. Q30 score on an Illumina MySeq means that there is 1 in 1000 chance of incorrect base call or a 99.9% accuracy rate. And cluster passing filter is the number of clusters that passed Illumina's chastity filter. A single molecule should generate a single cluster with clear signal. So a good MySeq run should produce clusters around 1000. In addition, the cluster passing filter score should be high, that is more than 85%, with a Q30 score more than 90%. So before loading the libraries on the MySeq, you need to place your reagent cartridge in water bath approximately 60 minutes prior to your run. Then HT1 buffer should be also kept in the ice bucket. After denaturation and dilution of the library, you need to pierce, the cartridges are foil covered. So you need to pierce the foil at position number 17 and load the library on the reagent cartridge. You need to wash your flow cell with milky water and wipe it thoroughly with lint-free wipe. And once cartridge is ready, load the flow cell and cartridge along with PR2 bottle in the sequencer. So MySeq pre-run checks flow cell setup, loading, reagent loading and setup. Please follow the guidelines provided by the Illumina during installation and training. So this is the flow cell slot on the MySeq, and this is the reagent cartridge, wherein this is the reagent cartridge and this is the PR2 bottle, which is incorporation buffer that is kept inside the sequencer, and this is the waste bottle slot. So this is the typical example of the library loading sheet, wherein you can see this is the panel name, that is source of the library. This is the nanogram per microliter qubit count of the library, and this is the average fragment size, which is determined using tape station. So after calculation using this formula, you will get 2.29 nanogram per microliter qubit count converted to 11.57 nanomoles. So for MySeq, it is recommended to convert your libraries to 1.25 nanomoles. So after converting this, you will get total, we need to add 1.73 microliter of the library to 14.27 of the 14.27 volume of the water to get total 16 microliter of the 1.25 nanomole library. So we need to first denature it using 0.5 molar NaOH. So during this process, because of the 0.5 molar NaOH, the libraries, double-stranded libraries will get converted into the single-stranded libraries, and after keeping that vial into the ice, because of the cold shock, the libraries will not re-anneal to itself, and it will become it will remain single-stranded till we'll put it inside the machine. So we also then after converting to 1.25 nanomole, it is further diluted to 7.5 picomole using HT1 buffer, which is provided in the kit. We also spike in the phi-x control during the run. So now what is phi-x? It is the adapter ligated library ready to use. It is derived from the small well-characterized phi-x bacteriophage genome, and it acts as a control for troubleshooting cluster generation problems to help determine whether an error is related to library preparation. So now let's move on to loading the libraries on the MiniSeq. MiniSeq kit also comes in two parts. Reagent cartridge is stored in -20 and flow cell is also stored in 4 degrees Celsius. Unlike MySeq, PR2 reagent is already incorporated in the MiniSeq kit, and MiniSeq reagent kit is also single used, and it is also provided with the RFID. So first, before loading, you need to first thaw your reagent cartridge in the fridge overnight, or you can thaw it in the water bath for 90 minutes prior to your run. Once thawed, the kit must be used within a week. Place the flow cell at a room temperature 30 minutes prior before to run. Then you need to thaw your hybridization buffer on ice and keep the flow cell for room temperature at least for 30 minutes. This flow cell comes in dry format, so before putting it inside the sequencer, you need to just wipe it through the 70% alcohol. So this is the flow cell slot for MiniSeq, and this is the reagent cartridge slot along with the waste bottle. So this is the example of library loading sheet for MiniSeq. So here also the qubit quant is converted into 2 nanomole library. And here also we denature the library using 0.1 normal NaOH. And for the recommended loading concentration for MiniSeq is 1.4 picomole. So after denaturation, we need to dilute it further to 1.4 picomoles. Now let's move on to loading of the libraries on the NextSeq. The flow of the process remains the same. First you need to thaw your cartridge, then prepare your libraries, then load onto the cartridge and then start the run. So for loading of the libraries, we need to first thaw your reagent cartridge in controlled water bath at least for 8 hours. You can thaw it also in the refrigerator for 12 hours, and you need to thaw it at a room temperature for at least 9 hours. So special conditions are to be maintained while thawing the NextSeq kit. So it is mandatory to plan your NextSeq run well in advance. So this is again one example of pooling calculator where your library nanogram per microliter we are converting it into 2 nanomoles, wherein library's plexity we are keeping at 1, and library size, which is determined from the tape station, is 350 base pair. So we are converting nanogram per microliter library to 2 nanomoles, because for NextSeq 2 nanomole library concentration is recommended. So this is the example of NextSeq loading sheet, wherein we are converting our library to 2 nanomole, and further we are diluting it to 350 picomole. In NextSeq, the denaturation of the library happens inside the cartridge using RSB with twin 20 buffer. So we need to prepare the consumables also for sequencing. We need to keep the flow cell at room temperature at least for 15 minutes. We need to thoroughly mix the cartridge by inverting it 10 times. Then we need to insert the flow cell into the cartridge and load 20 microliter of the library in the library loading reservoir, and we can sequence them. So this is the cartridge slot on the NextSeq, and this is the flow cell, and this is the cartridge. So in NextSeq 1000 there are no two different slots available for flow cell and cartridge. The whole assembly fits into the one slot. So now what is cluster generation? Cluster generation occurs on the flow cell. A flow cell is a thick glass side with channels or lanes. Each lane is coated with the lawn of oligos, which are complementary to the library adapters, and each cluster represents the thousands of copies of the DNA strand. So now there are in Illumina there are 2 different types of flow cells that are available. One is patterned flow cell and one is random or standard flow cell. In patterned flow cell there are billions and tens of billions nanovels that are fixed location across the surface of the flow cell. Each nanovel contains oligonucleotides that capture the prepared DNA strand for amplification, and it provides the even spacing for sequencing clusters. And in between the nanovels there are void spaces in which there are no oligonucleotides are present. So on that void spaces there will not happen the cluster generation. Cluster generation will happen only inside the nanovels. So because of which making the flow cell less susceptible for over clustering. In random flow cell, the clusters are randomly created on the non-pattern flow cells, and these are more susceptible for over clustering. Pattern flow cell you can see in NextSeq 1000, 2000, NovaSeq, and in random flow cell you can find in MySeq, NextSeq 550, MiniSeq, HiSeq. So during sequencing by synthesis, now what is during sequencing by synthesis? In each cycle there are 4 different fluorescently tagged DNTPs added with the help of DNA polymerase. The incorporated fluorescently tagged DNTPs will get excited, and then 4 images per cycle are taken, and using image analysis software the sequencer can find out which nucleotide is being added in the growing chain of the DNA. So in 4-channel SBS chemistry, bases are identified using 4 different fluorophores. Each sequencing cycle 4 differentially labeled bases are added to the flow cell. The imaging unit captures 4 distinct images using 4 different wavelengths. Image analysis software to determine which nucleotides are being incorporated. And with 4-channel sequencing, every sequencing cycle requires 4 dyes and 4 images to determine the DNA sequence. In 2-channel chemistry, which is used in MiniSeq, it uses mix of 2 fluorophores and creates 2 images. The clusters that are appearing in green are T. The clusters which are appearing in red are C. In both images, the clusters which are appearing are A, and the clusters that are dark are labeled as G. So after imaging, cluster intensities are plotted and bases are called accordingly. In two-channel chemistry, the real-time analysis becomes faster. NextSeq uses two-channel chemistry, but here blue-green chemistry is used. The cluster images are taken in blue and green channel for every sequencing cycle. So individual cluster intensities are extracted from the images and plotted against, and bases are called. So clusters with intensity green are only T, with blue are only C, and clusters with intensity in both channels are labeled as A, and dark cluster are appeared as G. So it uses blue instead of red to achieve greater resolution with its closely packed nanovels. So here are the different versions that are available for MySeq, MiniSeq and NextSeq kits. In MiniSeq kit, you have mid output and high output. Min output will give you 8 million reads in total, and MiniSeq high output will give 25 million reads. For MySeq, there are V2, V3, V2 micro and V2 nano. 4 different versions are available, having 15 million, 25 million, 4 million and 1 million read respectively. And in NextSeq, there are 2 versions, P1 and P2, which will give you 100 and 400 million reads. So for every sequencing run, you will also require a sample sheet. The sample sheet is a comma-separated sheet file that stores information required for the setup and perform the run. It can be created using Illumina Experiment Manager or manually using text editor or Excel sheet. It should be accessible on the instrument before the sequencing run. When the run begins, the software copies the sample sheet from the designated sample sheet folder to the output folder. At the end of the run, sample sheet is used by the instrument for secondary analysis, and FASTQ files are analyzed using bioinformatics pipeline. So this is the example of the sample sheet where you can see these are the sample ID. This is the workflow by which samples are prepared. These are I7 indices, I5 indices, and workflow labeled as generate FASTQ. Now, when you are loading any libraries that are not compatible with the Illumina, you need to spike in the custom primers. So spiking of the custom primer is necessary when your library design is not Illumina compatible. When one need to sequence custom library along with 5x or any other standard Illumina library, custom sequencing primers are added inside the cartridges. Designated wells are provided on the reagent cartridge for spiking the custom primers, and when custom primers are used for read 1 and read 2, the software directs the instrument to pull from the designated well on the reagent cartridge. So on each cartridge there are specified wells are provided to spike in the custom primers. So this is the MySeq kit example. This is MiniSeq, wherein 13, 14, 15 wells are labeled for custom primers, and in the NextSeq cartridge, this well 1, well 2 is specifically designed to add your custom primers. Thank you. Any questions?
Can you explain a little bit?
Custom primers mean the libraries that are not compatible with the Illumina read 1, read 2 specific primers. For that, you need to add the custom primer separately into the cartridge so that your custom library will also get sequenced along with the Illumina compatible library.
So this is only for if you are into developing of leukemia or whatever NGS methods. These are, as I showed yesterday, every Illumina library has Illumina sequencing binding motifs. That means I mentioned yesterday that inside the flow cell there are primers that come and bind to the standard Illumina library from where your NGS, from where the sequencing process starts, because without that sequencing cannot proceed. The primer has to bind, after which there can be addition of bases. So some ways in which we prepare the library, we replace the Illumina standard library, the standard sequencing motif, and we add our own motif. So if you have read some of our papers in leukemia where we did this for FLT3. Previously we have done this for NPM1 mutation. So those are ways in which you can incorporate sequencing, your own custom sequencing libraries. it has its advantages in the sense that you can get even better cluster generation, even better quality metrics as compared to standard Illumina libraries. But once again, 99% of people will not need to do custom sequencing stuff.
Thank you.
This is not similar to the custom. Just say you make panels for myeloid, lymphoid, everything. You have so many custom panels. So this is not for that, no?
So most of our panels
Are
do
in-house.
not require custom sequencing primers. Some like the FLT3 NPM1 assay we required. The MIPS assay requires custom sequencing primers. Rest of them do not.
Why don't they require so then?
Because we have designed them to be Illumina library compatible. So we have designed Illumina compatible libraries. So
So for the 5x adding, so we should take into account the diversity of the libraries if it is kind of a balance.
So 5x when you add is 5x number one. If you are doing some new experiment altogether, your own library you're making or something unique, Illumina will say that it is because of your library.
Means
If
I
you
have-
have
I have customized my panel.
so made your own thing, made your own experiment. See, the advantage with Illumina is completely open source experiment. You can do whatever you want with it. So if you have 5x there, it is a guarantee, or it is a way of evaluating whether the sequencing proceeded properly. Okay, so that is one way. The second is when you are sequencing low diversity libraries. So low diversity means typically if you are sequencing, let's say single amplicon. For example, you are just sequencing NPM1, you are just sequencing FLT3. What will happen is, because everything is binding to NPM1 exon 11, the entire flow cell will have just one base as the starting base.
Okay.
So everything will be the first base of after the NPM1 primer. Right? So as a result of which there will be sequencing errors. This is typically what is a low diversity library. So if you want to offset that, you have to add something which is got diverse bases everywhere, so that other signals can be read. So this is where 5x comes into play, because it is like, randomly you will get different regions of the genome.
So mainly for the single amplicon based the low diversity.
Low diversity. So low diversity, yeah, typically they will be single amplicon based libraries. Anything else I can think of, maybe 16s, but other than that I can't think of anything.
Thank you.
Thank you.
Thank you, ma'am, for your insightful presentation. Your guidance was extremely valuable, and I believe it has clarified many of the questions and uncertainties surrounding this important process. I now welcome Dr. Shruti Chaudhary, who will highlight common mistakes in NGS process and share valuable insights on how to avoid them, ensuring smoother workflows. Welcome, ma'am.
Good morning, everyone. Am I audible? So today I am going to present my talk on common mistakes and errors in next generation sequencing. So come to the basic workflow of NGS. Everybody knows that we do the DNA extraction, then we do library preparation, sequencing, and data analysis. But when we are preparing our library, every time we face a number of errors and the processing errors during that procedures. So if everyone says that I am going to make a error-free NGS library, it is really not acceptable. So if you put some necessary actions, that do's and don'ts in your mind, then only you can reduce the errors and you can get the proper results and you can release the results. Because when you are going to establish a NABL laboratory, your pre-analytical, analytical and post-analytical procedures should be proper, and that should be adequate and as per the guidelines. So for that errors should be counters, and that should not be happen in your system regularly. So based on the NGS workflow, we can divide these errors into pre-analytical errors, the analytical errors, and the post-analytical errors. So in pre-analytical errors, the sample preparation comes, And in the analytical errors, the library preparation means extraction to, you can consider the base calling and the quality check of the library. And after that, in the post-analytical errors, your alignments, variant calling in and your reporting, this comes under the post-analytical errors. Okay, so now we will discuss step by step altogether. So pre-analytical errors means the sample preparation. This is a very critical step. Every time we are not, every day-to-day work, we are getting these kinds of problems, but we are not taking account of all these things. However, this is ultimate important. That is the information error that can be a wrong identification, wrong container, sample is not adequate, it is insufficient, underclotted, or transported not properly. So all these things can affect your NGS results. So to avoid this information errors and all, the requisition should be made properly and the patient history should come to the clinician properly so that they can release the results, they counter check the results. Sample receiving should be very proper. So in our hospital, we have a system in which we can record all these things properly and maintain a data of all the samples. Storage of samples, not only at the time of receiving only, even when you are putting up backups of all the samples, you have to keep storage properly. And proper policy you have to make for the clotted samples also. Wrong containers are not acceptable. So heparin is not recommended as an anticoagulant for molecular testing. If you are sending a sample which is for RNA testing, it is better to send in the PAX gene tube. However, it is little expensive business. Cell-free DNA, if you are going to perform it, it is suggestive that you have to use the specific collection tubes so that you can use it and you can save your samples properly. Improper sample handling means this can affect the quality and the quantity of the nucleic acids and leads to the erroneous results. Now what we have to keep in mind whenever, because in NGS number of samples we are pooling all together and processing all together. So in that condition, avoid multiple samples processing all together, make a batch wise system and label one by one sample altogether. So we have a system in our laboratory that every day we are checking all the samples, whatever the processing is going on for lysis, for DNA and RNA. So this counter check, second person checking is utmost important. And obviously the sterilization of the bench tops. Now come to the analytical errors. It starts from the nucleic acid extraction. So if you are using not directly to blood, you are using WBC pellets, so your lysis is adequate. Okay, for in that case, your buffer should be adequate, that pH should be adequate. You can see here, this is a WBC there, but red lysis buffer is there. So you cannot accept this. The pellet should be very proper when you are getting after lysis, if you are getting it. Hemolyzed samples will not get you the proper results. Also, in our system, we are using a magnetic bead-based system for extraction. So this is a Promega kit that we are using it. You can see here the beads are there at the time of the final elution. So these beads, as per their concern, they are saying that it is not affected to the downstream application. But for NGS kind of things, we have to take care of these. So we just put all these samples into the Magna rack to reduce the to elute this thing, and we can get a proper eluted DNA and RNA. So these kind of things may hamper your results in a later on condition. If you are using RNA as your template one, so in that case the DNA contamination should not be there. Here you can see in this picture, number of samples the DNA contamination is there. This is the ABL housekeeping gene PCR, in which we have first extracted the RNA, we converted into the complementary DNA, and then we run this gel and we find it out. Here you can see the messenger RNA has not been converted properly here, only the DNA contamination is there. So sample is already degraded, you cannot get the proper results in these cases. However, these samples are okay. So for nucleic acid extractions, you have to take care at the time of lysis. If you are doing RNA extraction, use the DNAs properly. If you are using any kind of experiments in which you need higher amount of the DNA concentration. So in that cases, number of kits available in the market, so according to that you have to design your protocol according to that, in which you are getting a proper results, and you can select that kits and you can get a good quantity of DNA. Also the quality of DNA is also important. Also, when you are extracting your samples, means you are lysing or you are directly extracting RNA and DNA of some kind of samples, the baseline sample should be extracted separately, and the MRD samples should be extracted separately, because it can create a problem in your samples. You can get some contamination during these things. The second is the library preparation. So in library preparation, as Swapnali has also told you, that inaccurate pipetting is really a big cause in it, and it significantly impact the accuracy and reliability of your results. So every time the pipettes should be properly calibrated whenever you are using it. Non-calibrated pipettes can give you the improper results, especially in case of when you are loading very lower amount of the samples, 1 microliter and 2 microliter, if you have to add it and your pipette is not calibrated, then it can give you an erroneous results. So in that condition, you can use multi-channel pipettes, or you can use the automated liquid handling system. This is a little expensive business, but you can input your system also. Once we started our laboratories and all, we do the standardization protocols and we standardize the things. After that, we validate the things and we put it into the service. Service is going on, but suddenly after some time you are getting some problems during the time of your processing and all. So for that also you have to keep in mind that what kind of errors can be done. So in master mixes, the main problem is the contamination. So keep your pre and post amplification area samples and the master mix separately. Like some of the batches did library preparation yesterday, and they came to know that some of the work we did it in the PCR setup area and some of the work we did it in the post PCR area. So in that condition, you have to keep even of the cleaning beads also, keep separately in 2 different areas. Never be mixed all together. So maintain a unidirectional workflow, that is important. And if your reagents are degraded, PCR reagents, if your storage is not proper, then also your reagents are degraded. So these things really important in this factor. So this is a picture in which we are regularly use master mix and suddenly we are getting nothing in our this thing. So then we have to change the master mix. So we did check with the different master mixes with the different PCR cycles and all, because NGS is not a one-step thing in which you have to just put a PCR and after amplification you can get the results. It's a multi-step process. So every process you have to validate first and then only you can implement it. So you can see here, then we have to change it, and we have to change now this thing into GoTac master mix. So all these things can be happen when you are starting your service and this thing. This one? So here we had uses the hot start master mix and number of samples we had processed it, okay, but suddenly it was not giving us the proper amplification. So master mix was not proper, it was degraded. So then either you have a new aliquot of the thing and a new lot of reagent with you, you can check with that. Okay, we have checked with the other master mixes also, because every time when you have put anything in your service, it is necessary that you have to make a backup plan for that also. If something is not working, you have to be ready with the things. So at the time of the standardization and validation, it is better to go with that. So here we had checked with the different master mixes and all, different cycles and all, because in NGS not only that you have to do amplification. Amplification comes after WGS and all, if you are not working with the Amplicon libraries. So in that case also we can get here the results. So now I can have to choose it that which is the best one. Okay, then I have to do the whole library preparation with this, and when we do the tape station, we can get these results and with the interest of the band over there. So then only we can say ki we can proceed with that and we can move it. Okay, fragmentation. So under fragmentation means improper fragmentation. Fragmentation is like that you are cutting your DNA in a proper way. But if I interest to get a 300 base pair of DNA, it is not like that I am just putting a scissor over it and just cut it over there. So we have to make a number of standardization during time with the different type of temperatures. So then only we come to know that at which temperature we are getting our interest of gene or not. So here you can see my interest is around 300 base pair, but I am getting peak at 456. So this fragmentation is not proper, means I have to do some other temperature variations, and then only I can be able to get the results if whatever I want. So amplification cycle, this is also plays an important role in NGS. So usually low cycles are preferable to unbiased amplification. So for that also we have these results in which you can see some adapters are there. Okay, so amplification cycle, we have to keep in mind that at which amplification cycle we can get a proper peak and we can get a proper library, only then it is able to load it onto your surface. Cleanup of the library is the next step. Once the library has been made, you have to clean that thing, because number of adapters or unnecessary unwanted amplified products will be there and your master mix contents will be there, so that we have to clean up. So if you wish to keep anything, means like 300 base pair, so below that whenever you want to do the cleanup, at least 2x cleanup will have to do to remove the rest of the impurities, to clear these impurities. So here you can see these peaks having a adapter contamination. This should not be there, okay, because if this will be there, it will give you the downstream application errors. So in a summarized way, if I can say, you have to do for library preparation, the adequate quality and quantity of the sample should be there, the appropriate master mixes should be there, proper addition of indices. Whenever you are adding the indices, this is the utmost important step whenever you have to take proper care to addition of the indices. Pool barcoded samples in an equimolar manner. If as Swapnali has showed in his slide, that if you do not do equal addition of the samples, you will get results in a very different way. Some of the samples may get more reads and some of the samples may not. So equimolar amount should be important. Fewer amplification cycle can increase your efficiency of your NGS data. Bead purification, cleanup is at most important, and also addition of the control sample if you are adding, like if you wish to add OCIML3 for AML samples, we are adding it. So controls, if it will be there, that will easy to compare your results whatever you have get in that step. Minimize the handling steps means you can put automated systems for the extraction, for cleaning and all these things that can reduce the error of the manual things. So and the final and all that you have to follow the best practice for laboratory. Once the library has been made, then you have to do the library quantification. So quantification is the final step for sequencing run before, and it should be very accurate. If it is too low, you can if it is too high, you can get over clustered results and your passing filter will be fail over there. And if it is too low, you can get under clustered results and you can reduce your data in that case. So this is the under clustered picture and this is the over clustered picture, which you can find it out on the Illumina platform only. Okay, so for that, accurate pipetting and repeated freeze thaw reagents are not acceptable. Once you do the cleanup and all, you have to do checks with the qubit and tape station. As Dr. Nikhil has told you that qubit is used for the quantification and tape station is used only for the check of the quality of the things. So tape station is nothing but an advanced form of your electrophoresis only, but it comes in a peak manner. You can get a gel picture on the data also, but that gives you an idea that proper band is there, so library is according to of that size, so I can get a peak according to that ratio. Okay, so whenever you are using the qubits and all, they have provided certain standards. So one is the lower standard and one is the higher standard. So in that case, you can get a straight line curve over there and you can say your reagent is okay and you can start to do it. But it is important that you have to validate that data daily. Just like in CBC, we do the precision samples. We have to run a quality check over there. So what we are doing, we are putting a control DNA every day to check our reagents are proper or not. If our values are coming proper in that range, then only it can be acceptable that our reagents are not degraded, or it's not we can use it. Okay. And in tape station, if you are getting these kinds of peaks, the machine will not give you any results over there. Sometimes it may happen that you can get a very flat peak. It is there at 372, but the concentration is very low. But here you can see a proper peak over here, but some of the higher base pair fragments are there. So this could be the error of your fragmentation also. Now next to the loading of the NGS library, as I told you many times that we have to be careful at the time of pipetting and the storage of the reagents and all. But loading of library is depend upon the library type, the sequencing system and the reagent kits. So platform to platform, kits may vary, system may vary, and according to that you have to define it that which kind of library you are going to add it. Okay, so for that also the ultimate thing is that you have to load. If you are loading too much library and too less library, both are harmful for you. Library quantification is important. Based on that you can reduce these errors. The size of the library is also important. As Swapnali has told you that NaOH is used for the denaturation of the double-stranded DNA. So for MiSeq and Miniseq platform, we are using, we have to do this off system, and the concentration should be 0.1 to 0.2 normal. Okay, but in NextSeq it is the onboard. Now once the library is ready and we had load onto the sequencing stage, so what error can be done at the sequencing stage? So on sequencing stage, that can be an instrumental error, in which sometimes it may happen that delivery of the poor reagents and kits. It may be flow cell related issue, it may be optical system issue, and it may be instrumental errors. So one by one I'll discuss with you. So this is an issue in which we had received 80 MiSeq kits altogether, and we had run approximately 14 kits, and consequently we are getting the low cluster density from 117 to 580 only. This will create a problem in that. So for counter check, we have run another kit, okay, and with the same library, and we get a proper results over here with the 90% of passing filters. So we thought that this may be the error in the transportation. The kits are not transported well. So along with these kits, we have received some Miniseq kits also with the same temperature at the same time also. So when we run these kits with the samples, we can get the results also. It means that was not the transportation error. The reagents were transported properly. Then we had complained over there, and they have checked in their system that it was the manufacturing defect in the kits. So whenever you get another lot, this is a kind of lot verification. So you have to do it, and you can reduce the errors in that way. Also, we have received a defective cartridge of MiSeq, in which the cartridge was not fit into the MiSeq instrument. So before loading to the samples, means everything is ready and you are going to load the sample, and then you come to know that cartridge is not going inside, so it is create a frustration over there at that time. So the next is the flow cell error. So in this picture you can see the result of the Illumina. We had used the proper reagents, then reagents was not expired. The quantification was very okay, and we had load the samples, and we get cluster density of 431 only, and the run was failed at the step 3 only. So when we complained to the Illumina, they have checked the system and run the previous flow cell and find it out that this was the flow cell error, not to the processing error. Some optical system errors can be caused. This is all related to hardware errors, but it can be done inside the machine, or it can be done by our processing also. So this run failed due to over cluster of the library. However, you can see the cluster was there, passing filter and everything was there, but the camera was failed to capture the picture because there was too much library concentration was there. So camera was not picking ATCG properly, and due to the over cluster, the camera was failed to give the results. This is another some issues of optical system issues. In MISIC, one run was stopped at 279 cycle. Immediately the screen went black. So this was the error in the optical system. It was not error of the library preparation or something that we had made. This is again the issue like that. Instrument error. Sometimes it happens that they came to you update your system and all. So after updating control software, one run we had put of MISIC V3 and it was failed. So we have checked with the V2 500 and nano kits also. In this way we have wasted our 3 kits in that way, but ultimately the problem was there with their updation, and we had lost our kits. So we asked for the replacement for this. Another is the environmental issue. This is very important issue. When you are going to establish your laboratory, you have to keep in mind that the NGS instrument should be placed in a proper area. Okay, so dust accumulation, it happens with us also. The camera was there and it was the optics was failed in MISIC run. So we just put this dust free means sticky mats outside the room, so that dust we can reduce the dust area and we can put a dark room to clean and maintain the things. Also humidity control. Humidity control means as per Illumina guidelines, they are considering that the relative humidity should be between 40 to 70%. So in Mumbai like of area, humidity is really very high. So in that you have to keep, ideally that you have to put dehumidifier for this, but you can keep the ACs and you can make a dust free area and the less SS of that control area, you can reduce these things. Or for that also you can maintain a humidity chart every day. What is the temperature log over there and temperature monitoring and the humidity controller, that can help you to reduce these kinds of errors. So the next part is error is the which is related to the library and the reagents. So it can be of library design, quantification issues, under cluster, over cluster, and the expired reagents. So here again it can be the custom library, as Dr. Nikhil has explained you this thing. Quantification error, if there is an adapter contamination, then your results cannot be get proper. Over cluster, if here you can see it's a nano kit, MISIC nano kit. The cluster density was around 1393 K per oblique mm2. But the thing was that the range of this nano kit was 800 to 900 over. But we have get the cluster more than that. So however, everything was okay, but it is not adequate quantification. So we had failed this run. Even repeated freeze thaw of the Illumina kits can affect your things. So one times it happens with us that we had run a MISIC V3 reagent cartridge and it was thawed 2 times before the loading off to the library. Because in library it is whenever we have to load the library onto the kit, we have to thaw it properly because reagents are there inside the kit only. Okay, so but this is repeatedly freeze thaw manner. The run was failed with the error. But the next day we had run to check whether this is our error or the instrument error. The another library which was run on the micro kit with 500, the run was proper. So with and it was completed with no error. So then we came to know that this was the repeated freeze thaw error takes place at that time. Yes, yeah. So now the bioinformatic errors and mistakes. So the poor quality reads and the sequencing error can be considered as the platform errors. Okay, adapter contamination means the incorrect alignments of the adapters, and the alignment errors can be give you the incorrect variant calls. So all these things errors can be takes place at the sequencing level and the bioinformatic level. Now, another bioinformatics errors are the ambiguous alignments. So this is usually takes place in the short reads alignments, and for this you have to do the pair end assembly. Software version conflicts in which different version of the software or tool might be produce the inconsistent results. Annotation errors means incorrect biological interpretation takes place, and variant callers means some of the variant callers may not detect some of the particular mutations, so you have to add something extra. So I'm going to explain this thing. This is a variant caller. It's a result of an AML with NPM1 FLT3 ITD mutation, conventional PCR. So we did a capillary electrophoresis and we can see a 4 base pair insertion in NPM1, and this is the FLT3 insertion, which is get only by the get ITD. So in NGS results we can get the results in this way. So this is C in which this alteration has taken place in FLT3. So annotation error is the HGVS error. So here it's a NPM1 gene in which the C is altered with that 4 base pair insertion. So when we see in the IGV, it is insertion find it over here. So you can see TC, TC, TGG, but here between C and T 4 base are inserted. So according to the HGS nomenclature, the third rule, the insertion takes place at the 3rd base pair, and we can consider that this is the mutation. Post-analytical error, as I already described you, that this takes place at the post-analytical stage. So here is an example of BCL11B in which the variants were very less, but more than our requirement of the 5% VAF. But here you can see all the filters which we had put the variant callers, they have rejected this thing. So when we have checked the data, you can see here the multiple backgrounds calls are there, and sometimes C is changes to T, and some of the T bases are also there. So it is suggested that this is a sequencing error. However, you can see here it is a proper true calling, true mutations, true variants. Here A is going to change in C in all the things. Here is an example of KIT mutation in which okay, no problem. It is a mixing of some NPM1 NRAS mutations in which all these mutations seen in number of cases, but when we do the extractions, we get a clear results, and we can say that that was the contamination in the DNA extraction. So the take-home message is that whenever you have to do, you have to improve the efficacy of the NGS experiments, you have to perform pilots runs and replicate experiments, randomize of samples, and orthogonal method validation. Thank you. Any questions? Okay, thank you.
Thank you, Dr. Shruti. Your guidance on avoiding these pitfalls will undoubtedly help us achieve smoother workflows and more precise results. Next, we have Dr. Gaurav Chatterjee, who will provide an overview of long-read sequencing, exploring its unique advantages and potential applications in genomic research.
Okay, so we'll start with the problem. So as you can see, this is a 13-year-old boy, and you can easily make out this is the case of BALL, right? So all the BALL markers are positive. So the question is that what investigation typically you will do in your lab for this child, and in what sequence? So the answer to that at current time point, what we do in our lab is that we do a series of investigations, which is usually followed in a sequential fashion. So we do a routine FISH and RT-PCR. And if they are negative for that, then maybe you'll go for a targeted RNA sequencing to detect all these different types of fusions. If they're negative for RNA sequencing, then maybe you'll go for a DNA sequencing, which can pick up some of the sequence mutations like PAX5, PATR. So, but as you can understand that this process is sequential and this takes a lot of time. So could there be a test that can replace all of this and that can detect all of these problems at one go only? So the point that I'm trying to impress upon you is that current sequencing assays, which are short read based, are often inefficient and incomplete. It requires stepwise testing, multiple clinic visits, and sometimes even multiple sample collection, which causes lots of time delay in the diagnosis. So this is a different type of a problem. This is problem 2. You can see a 3-month-old full-term infant who presented with an infantile encephalopathy. So the problem here is that this has a huge list of differential diagnoses. Almost 1500 genes could be implicated here. So, and also with each passing moment, the child is incurring permanent neurological injury and death is ultimately inevitable. So if you really have to attempt a genetic sequencing in this case, what test could it be? And what is an acceptable TAT here when every minute is important for the child's life? So with these two problems in the background, we start our topic today, which is on long-read sequencing. This will be slightly different from what we have been listening to so far today, which is more practical. This is going to be a little bit of chemistry, a little bit of applications, and hopefully some of the things, some of the things how future is going to be shaped up. So this was I think published 3 years ago in Nature, the milestones of genomic sequencing in 20th century, and one of them was long-read sequencing. I encourage you all to actually go through all this. This is a very nice kind of an historical summary of what are the milestones that was achieved in genetic sequencing in the 20th century. So if we very simply have to understand how long-read sequencing is better in aligning in solving the genome jigsaw, so to say. So let's say this is a WHO hematolymphoid 5th edition book you have, and I give you these snippets, like for example this 4 and 5 words from the chapter of CHIP, and I tell you that stitch them back together and make me the whole book. So maybe you will be able to do it because you are of course learned, you know these topics, but still there will be some gaps probably if you just given these snippets, right? Or whereas if I give you paragraphs like this, if you get whole paragraphs at one point of time, of course the stitching back is much more simpler. So this is how long read is more beneficial than short read. For example, if you see to construct the human genome at 30x coverage, you will require 100 times less reads for PacBio Hi-Fi and 500 times less for Oxford Nanopore. So that amount of easier it is to reconstruct the genome using long read sequencing. So, and again, some of the problems of short read is not only about aligning, there are other problems, like there are some dark regions of the genome, which is almost 10% with short read sequencing, which usually has high repeat region or high GC content or segmental duplication. So you could see one of the examples here. You can see that this green, yellow and black are unique sequences, whereas the blue here are some repetitive regions. So because you will have spanning reads like this, which have sequences from both parts, you know that you know the spanning, but because the blue is repeated, so you know that yellow is upstream of blue, you know that black is downstream of blue, but the green could be both upstream and downstream, right? So if you have to stitch them back together just using short read, it could be problematic. Whereas in long read, when you have the full thing in one read, it's much easier to align that. And therefore, the short reads are problematic for detection of large structural variation as well, which can be detected using long read sequencing. Another problem is haplotype phasing. So this is very critical in disorders which have maternal or paternal imprinting, like Angelman syndrome or Prader-Willi syndrome. So for example, if we have to know in short read, you will know that there is an A here and there is a C here, and there are different regions of the genome which is either blue and red. But you cannot phase it. You don't know A is with blue or A is with red. That you will never know. Whereas in long read sequencing, because you are having the whole thing in a single read, you can phase it. You know that the A is with this blue part of the genome, whereas C is with the red part of the genome. And then finally, the GC content is also a problem with short read sequencing. You can see that as the GC content increases, the short read sequencing efficiency reduces, whereas long read can detect that as well. So in today's thing, we will just focus on the two main long read sequencing players, which is PacBio and Oxford Nanopore. Like Shweta mentioned yesterday, Illumina and 10x did come up with their own long read technology, but it was not true long read. It's what is known as synthetic long read, where you tag a long read of the DNA, but then you do the similar short read sequencing and stitch them back together. So these are not true long reads, and they never really came up to the same level with PacBio and Oxford Nanopore in this space. So we'll focus our talk only on these two. So just before that, just what are the common things here? So both of them are single molecule real-time sequencing, although the PacBio thing is known as SMRT, but both of them are SMRT. They routinely can produce 10 kb plus read lengths. The read accuracy has now started to approach Illumina benchmarks. This is very important. We will discuss about it. As we've discussed, the dark regions of the genomes are accessible, and therefore they are better suited to detect the structural variations. And one more important thing is that because it is single molecule real time, they are reading native DNA as opposed to amplified portion of the DNA or captured portion of the DNA in Illumina. So what that means is the modifications of DNA are also preserved, and you could actually detect them. So the epigenetic changes can be detected in the same run, which is again a massive advantage of long read sequencing. So with that, we now focus a little bit on the chemistry of long read sequencing. So this is PacBio. So the history is quite interesting actually. So this is James Corlack, who was a graduate at Cornell lab at that point of time, and he was fascinated with the structure of DNA poly. So we know that DNA poly is highly evolutionarily conserved across species. It zips through the DNA 100 base at a time. So he wanted to just see DNA poly in action. So this thumb and finger-like configuration that you are seeing here, how it holds the DNA and how it adds each nucleotide. So all he had was a curiosity to see DNA poly in action. So he collaborated with his friend Stephen Turner, who was in a neighboring lab in Cornell, and they then came up with this technique. So we'll see how they managed to do that. So at the heart of the library preparation is this structure called SMRT Bell, which is basically a double stranded of DNA, which is added with a hairpin adapter, couple of hairpin adapters to make a circular structure. And then finally you add the primer and DNA poly here. So this is the complete SMRT Bell structure. So then this is the machine, this is the Sequel. They have the Revio now, which is the updated version. And Sequel has 8 million and Revio has 25 million of these SMRT cells in this region. And so how is that achieved? How you can see DNA poly? So the key to that is this structure called ZMW or zero mode waveguide, which are present in this SMRT cells. So they are extremely small. So we are talking about zeptoliters of volume. Zepto means 10 to the power minus 21. So why is that important? Is that what they managed to do is that the DNA poly is immobilized in the bottom of the ZMW using streptavidin. And then what you need is light to hit only this portion and not anywhere else of the ZMW. So how was that achieved? The diameter of this is so small, it is even half of the wavelength of light. So according to the laws of physics, light will decay exponentially as it tries to go up because the diameter is so small. So light will only hit this bottom portion of the ZMW. So then what happens is that you have a sea of this fluorescent tagged nucleotides, and at one point of time only one nucleotide is being added with the chain, and that is the point of time that light hits that nucleotide, that fluorescent fluoresces, and you detect that signal. So at one point of time only one ZMW contains only one DNA poly and one DNA molecule, and then all the colors flash at real time. So this is how they could manage to see DNA poly live in action, one molecule at a time. So this is the output that you see. It is like a movie. You see this kind of colors that come out. One more important thing is because it is real time, so the kinetics is also important. So it's not only T, C, G and A. You could also see the duration between each call, right? So this is known as interpulse duration. So based on that, you could make whether there is epigenetic change or not. So for example, if there is a gap, the longer gap, before the upcoming T incorporation, this you could make out this is the 6-methyl adenosine changes going on. So this is known as interpulse duration. Okay, so this was the technique, but the problem with this was that this error rate was quite high. So it was almost 8 to 15%. As I told you, that light needs to hit only at the bottom portion. So it's possible that some of the nucleotides which are not being captured are also getting fluoresced. So there would have been some error rates. But what they realized, the real game changer in this technique, I know the term game changer is used quite loosely in the space of genomics, but here it was really game changing for PacBio. So what they realized is that the structure is already circular. So if I compromise on the read length a bit, and the DNA poly life can be extended a bit, so the DNA poly will go on and sequence the same region again and again, right? So this is the circular structure, and it has sequenced the same region of DNA again and again, and therefore you can have, similar to the error corrected sequencing approaches that we use in short read, you could have a consensus read which will clear out all the random error. So this came to be known as the circular consensus sequencing or high fidelity sequencing, quite dramatically then later on named as hi-fi sequencing. So this PacBio hi-fi sequencing can approach a Q30, that is 99.9% with 10 passes. So means a same region of the DNA read 10 times. With 4 passes they could reach a level of 99% accuracy. So at today's date, the most comprehensive and the most accurate genome remains to be PacBio hi-fi genome. So if you have a child with some developmental problem, you do not know what it is, and you really want to see what it could be, the PacBio hi-fi genome seems to be the most comprehensive and most accurate genome. So that was about PacBio, and let's come to the chemistry of Oxford Nanopore. So this is a read length agnostic technique, meaning that in principle the read length can be unlimited. So the both of the strands are attached to this motor protein and adapter tag DNA. The function of this motor protein is that it has a helicase and polymerase activity which ensures tethering through these nanopores which are present in the membrane. This is the structure of the nanopore. You can see that the motor protein has tethered to it, and the DNA has unwound, and one of the strand of the DNA is passing through the nanopore. Sure. Sure. Okay, fine. So I was just talking about the chemistry of Oxford Nanopore. So you have this adapted motor protein and adapter tag DNA at both of the strands. The function of this motor protein is that it tethers the DNA through this nanopore. So what happens is that we have a current difference between this nanopore, and because DNA is electrically charged, it will pass through it. But you have to ensure that the DNA is coming to this nanopore. So for that, the motor protein is there, and also it has a ratcheting mechanism, meaning that it ensures that only one directional flow is possible. It's not going to come back. The DNA is not going to come back. Of course, the current difference is also ensuring that, but that is the function of the motor protein. So this is how it goes, and the chemistry over the year, how it has improved is this nanopore structure has improved. So if you see, it initially started with alpha hemolysin. Now the current chemistries use this CSGG thing. So the fact is that the sensing region has reduced. I'll come to that why it is important. I think Karthik, you asked a question yesterday, so it is related to that. We'll come to that in the next slide, I think. So this is the minion. You can see that this has all this DNA tagged with motor protein adapter sequence, and how it goes to this tethering structure and it goes to the nanopore, and this is how the sequencing takes place. The DNA is basically passing through this nanopore because of a current difference between the two surfaces. And how it is read. So this is what I was talking to you about, Karthik, yesterday, that a KMR sequence is read together. So what it means is, it's not only one nucleotide. We are reading this 1, 2, 3, 4, 5, 6, 7, 8 nucleotide at one point of time. So the smaller you can make the sensing region, the more accurate your reads are going to be, right? So the current chemistries have come up with smaller sequencing ports. And finally, this changes in current are read as a squiggle plot. This can be compared with the notes of music. It is like the notes of music, how the bases are changing. Based on that, we read them. It's very unique. It is completely electrical. So this is the only sequencing technology that uses electrical technique to detect the changes. Not any kind of optics is involved here. This is the different chemistry. I'm not going to detail. So current chemistry is known as V14, which has both the strands of the DNA are sequenced. And like I told you, the scanning region has reduced, and now this R10 chemistry actually uses 2 of the scanning ports, so thereby improving the accuracy even further. So right now they claim a simplex read accuracy of Q20 plus, and even with duplex read accuracy it could be Q30 plus. But still, at today's state, the PacBio hi-fi seems to be more accurate than Oxford Nanopore, but they are still approaching an accuracy rate of 99% with these further chemistries. 2 kind of read lengths have been classified as long and ultra long. So ultra long means even more than 100 kb. But again, because in principle it is unlimited, the read length, so it basically depends on your ability to process DNA with that read length, right? So if you change the processing technique, you could actually achieve even higher length of DNA probably with this technique. The highest read that has been so far documented is more than 4 megabases. So that is the amount length of DNA we're talking about that can probably be achieved in extreme situations with Oxford Nanopore. So these are some of the flow cells. The reason I'm showing you is because of this, the Flongle, which is quite versatile, which generates only 2.8 GB of data, but it can be, it's very quick, it's most accessible, it can be used anywhere. So this can probably ensure like a bedside sequencing, something like that. And then you have Minion and Promethean flow cell, which produces more data. So just a little bit on the targeted sequencing approaches on Oxford Nanopore. So you can, in long read, you can use all the normal target sequencing approach as well, like PCR or hybrid capture. Although it will compromise on your native DNA reading ability of the long read. You can use CRISPR-Cas9 based approaches as well, but what is really unique is something known as dynamic time warping or adaptive sampling. So this is a completely computational approach of target enrichment. I will say that again. So there is no chemical modification at all. It's completely computational approach. So how does it happen? So in Oxford Nanopore, as I told you, there is a current difference between the two surfaces, right? That is why the DNA passes through. And also the fact that it reads real time. So what is happening is the DNA starts passing through, you read it real time. From the initial basis, you understand that this is my target portion. So the DNA sequencing goes through. From the initial reading of the basis, you realize this is not my target portion. I don't want it. So you just reverse the current between the two surfaces and the DNA is ejected. So it's a completely electrical, completely computational, without any chemical approach of target select enrichment, which is only possible with Oxford Nanopore technique. So just to summarize the two things, the PacBio basically is a direct visualization of DNA poly in action. It's using optics. It's like a movie. Oxford Nanopore is changes in current, which is read as squiggle, which is like a music. The read length is typically shorter for PacBio. As we have discussed, there is a compromise. There is a trade-off between read length and accuracy. The lower the read length, the further accuracy will increase with hi-fi reads. Whereas the Oxford Nanopore read lengths are principally unlimited. They can reach 100 kb using the ultra-long Oxford Nanopore. Hi-Fi seq is more accurate at today's date. Q30 is achieved with 10 passes. The Nanopore is approaching a 99% accuracy, but Hi-Fi seq remains to be more accurate so far. The maximum output with the current machine Revio is 360 GB, whether it stands at a staggering 14 TB with Promethean P48. Of course, you then will have to use all the flow cells together, which is probably not going to happen. But theoretically, this is the maximum output of Oxford Nanopore. In general, PacBio is costlier. If you look at per base sequencing cost, it is more for PacBio as compared to Oxford Nanopore. And the Oxford Nanopore provides the unique flexibility of fungal and on-field or rapid sequencing. So this is just to summarize the differences between these two technologies. So now we'll see just few of the applications of long read, how they are goes through. So the first is large structural variant or tandem paper. We have discussed about that. So this is a paper that compared the techniques, and they found out that almost 47% deletions and 74% insertions were actually missed by Illumina whole genome sequencing, even with 11 variant caller. The main reason is that these large variants cannot be picked up with short read sequencing. Those are only picked up using long read sequencing. This is an example, I think, where a novel GGC repeat was picked up in this gene in a child with leukoencephalopathy. So these are various kind of applications are there where the structural variants can be picked up much more easily using long read sequencing. And some of these structural changes are not only disease causing, they make us what we are, which is human. So if you look at this gene expression plot, some of the genes that are mostly different between human and our closest, closest colleagues in the ape genera, so the chimpanzees, that is. So you can see that some of these genes which are mostly different in us, the mechanism of that gene difference is some structural variation. So long read sequencing not only picks up disease causing novel structural variation, they also tells us why we are unique as a human. So what are those structural changes as well? So those are also possible using long read. This Nikhil sir has showed yesterday. This is the fact that de novo genome assembly is much more easier using long read. This is a plot I'm showing you of 2019, which was HGP 2001 versus the telomere to telomere consortium in 2019. So this alternate gray and black portion you can see are known as contigs. It means that those regions of sequences I know for sure. Then there is a gap and then there is an alternate contig that way. So you can see that number of contigs that were there in 2001. So that means that number of gaps that were there in 2001. Whereas in 2019, X was gapless. This was the first gapless chromosome to be sequenced. The other chromosomes still had some gap to be filled. And in 2022, this plot Nikhil sir has already shown that we now know except chromosome Y, all chromosomes are now gapless. And this black and red portion that you can see, these are you controlling? No. Are you controlling the mouse? No. Okay. Sorry, it's moving. I don't know why. So this black and red portion that you can see are the sequences of DNA that has been introduced using a long read DNA. Anybody can tell me why chromosome Y is not sequenced in this? Yes. So Nikhil sir has told you this is the complete heredity from mole, which is 46XX. It doesn't have Y chromosome. That is why this was not. It remains to be sequenced. And then finally towards the human pangenome, again this has been spoken about. So now we will probably look at a future where we do not only have one reference genome to align to, but major genomic variations as well. And again long read sequencing will be instrumental in detecting this, developing this human pangenome. So coming to the problem one of the problems that we started to. So what happened to this child? So this was a 3-month-old child. She was subjected to an Oxford Nanopore sequencing and they could pick up a CSNK2B variant within 7 hours of time. And therefore this CSNK2B related disorder was diagnosed and disease specific treatment could be initiated. Another spectacular example has been documented in thiamine metabolism dysfunction, although this is with short read sequencing. This was I think within 16 hours they could do it. But that took a lot of computational collaboration. Whereas in short read you can see that this is the paper that was published. They showed that within 7 hours they can routinely give you the result. So for this kind of ultra rapid genetic diagnosis, when it is needed, the Oxford Nanopore based long read sequencing seems to be a viable answer. And then moving beyond DNA, like we said, that the combinations are difficult to understand with short read, right? So let's see this hypothetical example of a RNA which has couple of transcription start site, couple of alternate exons, one SNP here, then the standard exons, and couple of poly A site. So all these possible combinations can be there. So if you do just short read sequencing, you are picking up all the spanning region, that's fine. But then as a whole, all these possible combinations could be there. Whereas using long read, it was known that only this 4 was truly occurring in human body. So all these isoforms of RNA, the full length isoforms of RNA, can only be probably known using long read sequencing. And one more advantage of Oxford Nanopore is it can also detect native RNA. So PacBio, you still have to make it cDNA, but for Oxford Nanopore, they can even sequence native RNA. So all these RNA modifications are still accessible. So that brings to the next application, which is, like you have said, epigenomics and epitranscriptomics. Epitranscriptomics means the RNA modifications that is also detectable. So how do they do that? We have discussed in PacBio, it is the interpulse duration based on which you can detect. Whether in Oxford Nanopore, it's simply a change of current, which based on that you can detect that there is a change going on. So in the same run, you are detecting epigenomics and epitranscriptomics as well. Another application is in metagenomics, which I'm not touching, which I think Rahul and Arpit will speak about on that. A spectacular example of this is actually published a couple of years. Sorry? Yes.
How different is that from the chipstick?
In terms of what you was asking?
You said the gaps will tell you that.
Correct. Correct. Chipstick is a technique that can be used with this.
That uses shear stress to that.
Yes.
So the gaps may be corresponding to the.
Correct. It is also used for those linkage analysis kind of thing. But again, you are compromising on lots of more informations. Like in a single read, can you detect all the changes that you want to detect? So here you have a long read where you're detecting sequence mutations, but you're detecting all possible isoforms. You're also detecting the assembly. So you are right. The chipstick and Hi-C was also used in the T2T consortium along with optical mapping and other technique. But the long read couple of techniques are most useful in fulfilling the gaps of the genome based on that. Because
Chipstick is another separate library prep altogether.
Yes, that is also true.
Here
yes.
in a single experiment, you can get sequence level information as well as de novo, as well as in situ methylation status as well. Yes. So those sort of changes you will not get with Chipstick.
Yes.
Because that is a separate experiment which requires bisulfite treatment.
No. Actually, I had a slide I deleted for sake of time. Not only methylation, so it's a huge, actually maybe I could show you sometimes, but I could show you in the later on. So there's a huge list of changes that can be detected by this change of current, which is much more than what we could detect initially. Oxford Nanopore detects more because this is just current change. PacBio can also, especially this 6-methyladenosine is very useful. PacBio detects it much more efficiently, but Oxford Nanopore detects much more spectrum of these changes.
Why is that? Now in Oxford Nanopore, it's quite clear that when there's a methylated thing, there is a change in the.
Correct.
So you get a different call.
Correct.
Like a call is very direct.
Correct.
Why there is a duration delay in PacBio with respect to a methylation?
So it's real time, right? So we are seeing the DNA poly in action real time. So if the DNA that is it has been sequencing, it has a modified DNA. It takes slightly different time to take the next nucleotide and to attach it. So by that change you can do it. The key here is that it's real time happening, so you can understand that kinetics.
Because of the histones that are bound to the DNA and just to traverse.
Correct. But then if it is not real time, you cannot detect that, right? So ideally, even with a native, if you do a native sequencing in Illumina, then you could have detected it, but you cannot because it is real time. So the kinetics has to come live for you to know that duration. So this is a spectacular example of this ultra rapid epigenomic. This is brain tumor we are talking about. This was in 2021. It was published. So say they did brain tumor methylation status within 91 minutes, within like 1.5 hour to 2.5 hour, and frozen section like intraoperative precision was made based on that. So even that type of ultra rapid testing is possible using Oxford Nanopore rapid sequencing. So finally, we'll finish with few challenges and what we wish maybe from this technique so far. So first is reducing error rate even further. We know that PacBio hi-fi reads are consistent in read length and they have a higher accuracy as compared to Oxford techniques so far. This is 2020 plot, I think. So then Oxford techniques have improved in their accuracy, but still we will want them to further improve their error rate. So to be really there with Illumina benchmarks. Then the most important thing is we want the cost to lower down. So if you look at this, theoretically, the cost of a 30x human genome is 5 times using Revio as compared to NovaSeq X, and 3 times using Promethean as compared to NovaSeq X. So when as the cost comes down, we'll be able to do more and more with like gene with low expression levels, small amount of sample, rare event analysis like MRD and ctDNA. So this field is still to be explored using long read, although there are some circulating tumor DNA papers have started coming. But as the cost comes down, this will open up. And finally, I have not discussed about bioinformatics of long read at all in this talk. So they need a separate kinds of tools, separate kind of familiarity. And if you want to truly democratize sequencing, you will require more automation. So PacBio, to that end, they are now collaborating with Google Deep Consensus, and I think it is integrated in their Revio system as well. So those kind of better automation will be required to make it even more democratic, those sequencings. And finally, what we wish is a one-box sequencing, right? We do not want to go through all these sequences, all these stepwise decisions. We want a single test, single long-read sequencing that could detect all of this in a manner that is faster, more accurate, longer, cheaper, and can be performed anywhere. So from this anywhere point of view, Oxford has recently come up with a concept called Oxford Nanopore toothbrush, where you probably can sequence using a toothbrush every day, and like maybe a patient of leukemia can know that when the disease started to come back using just this toothbrush. So maybe that kind of future also we are looking at. So this is my last slide. Long read sequencing holds a lot of promise. I hope I have managed to convey that to you. This was Nature Method of the Year in 2022, and the editor wished that maybe someday we'll have reads that will be sequenced at DNA, RNA level, and with complete genomics, transcriptomics, and epigenomics at high throughput and accuracy. So with that, I end my talk. Thank you so much. Any question? Yes.
I read many papers on ONT, sir.
Yes.
So I came to know that there is some problem with the wax. Basically, if we are going through the block, sir, if we are doing the liquid part, that is okay with the ONT.
Correct.
But many of the people, they are facing problems with the
Correct.
wax, sir.
Yes.
So is there any solution?
So I think the recent that R.10.4.1 they have said that specifically looks at this as well. But I agree with you. It remains to be seen. The error rate is higher in that in tissue, and we require better chemistry to improve upon that.
It's very sensitive to degraded DNA. I think the quality of input DNA is most critical for these long read sequencing processes. As Gaurav said, the R10 is better simply because it enables duplex sequencing, and therefore the accuracy of sequencing is better. So it's not to do with wax, it's the fragmentation of formalin.
So what I came to know, sir, that the wax clogs the pores, sir, and the times the...
So impure DNA. So we are seeing this in stool samples as well. If there is impurities present in the DNA, it will block the pores, and suddenly it will show pore availability drop down.
Yes. Thank you, sir.
Thank you. Bottleneck.
He will come across something or the other in every patient.
Correct.
How will we decide which has to be intervened upon and which is...
Absolutely. So that's a great question. Actually, I deleted that slide. So there is another wish list is this human pangenome type of thing. So we need better control databases. So because we are picking up novel things, there are, like I told you, some of the structural variation are present as a human. So we need to know what are normal and what are not. So we need much more comprehensive, better control databases before we start, Now what we can do is you have a specific phenotype of the child or of the person, and you then diagnose something, that's okay. But when you are looking at genome scale changes, then you definitely need better control databases to make that call.
Most of the current ones are based on short trace sequences.
Yeah, but the T2T, as we have discussed, has many parts of the genome that was filled with the long read only. So I don't think they separate it out like that this is long read sequencing, this is short read.
And pan genome, no, they are long read, but it doesn't matter whether it's long read genome or short read genome. Genome, if you see the text file of the genome, it goes on and on till the gene gets over. How does it matter?
It matters if you're trying to find something in the population which would be of sequence.
So you will get sequence variation, but it's not that somebody will have an additional gene.
Like what we're currently seeing are individuals who are sequenced using a short read technology.
Right.
And the variants, whatever we found on that. Suppose if you do a million using a long read.
Sure, maybe. Means at least for CNVs or structural.
For CNV, like we have said, so correct. So for structural variation, you need a better control database, surely. But otherwise, as a genome sequence, you just need to fill the gap. So which long read has done so far. Yeah. That is adaptive sampling, a specific type of target enrichment that can be done, which is not in routine Oxford Nanopore. It's a thing you need to do to enrich your target. It's a strategy which is
not applicable as of now.
It's applicable. You can use it, but it is not routine. So it's like, for example, you can use whole genome sequencing in short read. You can target enrich as well.
In real time, we'll see that this is not something
Yes.
we want. And
Yes.
So to give you an example, this is done using a bed file as to what you want to enrich, and the ONT will work there. So we're working with St. Jude's on this.
Yes.
They say using adaptive whole genome sequencing, within 36 hours of leukemia receiving the sample in the lab, you can give an end-to-end report. So if you ask me, this is going to revolutionize all kinds of diagnostics, at least for leukemia. You can not only detect translocations, you can detect ploidy, you can detect chromosome gain losses, you can detect even FLT3, you can detect TPMT.
That's the thing.
Cheaper, it should be in the ballpark of still 15-20,000 because but the scale of the whole genome is huge, right? You need to still do so many regions, and it's not cheap. I mean, not.
I think
cheap
he's asking adaptive sampling versus a whole genome.
Oh
thing.
yes. Sure. Because you can do one sample in itself with relatively low cost. So this is done only on promethion. It doesn't work on your Minion or this thing. You require a promethion for that.
Yeah.
The Minion, most likely Minion and Flongle. So Minion and Flongle is. You can even do human stuff on it. So we are working with all different centers in India to do this, to do targeted RNA sequencing for ALL using that same machine. Yeah. It's very cost effective.
That is R10, so that is compatible with all systems, not only Promethean. Yeah.
The main problem that we are seeing is that the Flongle has a lot of quality issues even now. So it's not reliable, but Minion is more stable. So the poor quality drops fast. Still, they haven't ironed out few of the chinks there. But Flongle is phenomenally cheap. It's 7,500 for one flow cell. You can put 2 samples in there, and you don't need lot of upfront investment. So. Fast, no need to spend on. See, you have for any short read thing 1.5 crore or 1 crore. You need to set up a whole lab. Here, you don't need to do anything. Just take that thing, do your PCR and get started with it. You don't need compute infrastructure. You can just connect it to your laptop.
Yeah.
So it's very, very cost effective to no upfront payment. And plus routine sequencing is also very cheap. What he didn't show was that there is something unique about Oxford Nanopore. You buy one flow cell of the Minion, you use it today. अच्छा मेरे 2 sample है मैंने run कर दिया. You remove it from the flow cell, put it in the fridge, remove it 4 months later, still use the flow cell till the things are used using a wash kit. So this is absolutely spectacular, where you don't have to actually exhaust the whole flow cell. You can keep using it, washing and using, washing and using. We are going to set this up across 5 centers in India. So let's see how that goes.
Right. I have met Rahul for so long.
Thank you, sir. Your discussion on the unique advantages and potential applications of long-read sequencing in genomic research was extremely informative. Next, I call upon stage Mr. Rahul, who will discuss the application of Nanopore 16S sequencing in studying the gut microbiome.
Good morning. I will not take much of your time. I know everybody is waiting for the tea break. But please bear with me and apologies in advance because I have lost my reading classes 2 days ago. So, yeah. Please ignore my slips. All right. Most of the heavy work has been done already by the previous speakers, especially Dr. Gaurav, Dr. Swapnali, Dr. Shruti, and even previous days' talks. So they kind of do most of the heavy work for me. So I'll just quickly jump onto one of the applications of Oxford Nanopore, specifically for the gut microbiome. Now, how do you understand gut microbiome? And we know, again, it is coming up to be very important in all kinds of things. It modulates our immune system, it helps in the development, function, and even the gut-brain axis. Everybody is probably aware of it. So how do you get to know the microbiome? It could be anywhere. It could be on the surface, inside your gut, it could be in the soil, wherever. You do something called bacterial 16S amplicon sequencing. Now, for that, you need to focus on a particular region, which is sometimes called a marker or a barcode for that organism. It's exactly like how there's a barcode region for a particular product in your supermarket store. Just like that, there could be a barcode sequence or a marker sequence for every species out there. And that kind of thing is enabled because you have this 16S region in the bacteria, which has several conserved and variable regions. Now, they are conserved enough so that you can differentiate within a species, but variable enough so you can differentiate 2 species. So if you have a database for, let's say, all the species of bacteria out there, all you have to do is, sequence these cassettes in each of the species in your sample, put them in the database, and voila, you have your list of species in the sample. So that is what we kind of want to do. And you can also do it with Illumina, you can do it with Nanopore. We will look at the Nanopore context. So for the Nanopore microbial 16S sequencing, what you do is, first, you get your sample, you extract it, you extract the genomic DNA. So you will end up having a soup of different bacterial genomes in your elute, in your collection, in your extract. Then, you can do it with the Promega system, or you can do it with something else like the QIAGEN, or let's say Zymo research. People have their own methods. Once you get the genomic DNA, you end up doing the amplicon PCR. Now, why are we doing this 16S amplicon PCR? Can anybody tell me from yesterday's talk, how long is the human genome? How many letters were there? 3 something? Billions, right? 3 billions. Can you guess like the average bacterial genome size? Let's say E. coli. Quick guesses. Anything, whatever comes to your mind. In MBs, so I heard somebody say in MBs. Any guess? Sorry? So I'll just put it out across. It's about 4.6 megabase pairs, an E. coli. So if you want, we could, again, Oxford Nanopore allows us to do these long read sequences, but we have time constraints and we have resource constraints and we have all kinds of other constraints. So we can narrow down to this marker region. That is what makes it more rapid. So this 16S amplicon is nothing but 1.5 kilobase pairs long. So you do not have to sequence everything else, you just focus on this region. And so the primers will come and sit on all the 16S regions of all the genomic DNAs in your soup. And then you then prepare the library, you attach the adapters, you load them on the sequencing, the flow cell, and then you do the bioinformatics. Just to give you a quick overview, the way you can process the sample can also affect your downstream data, homogenization of the tissue, whether you are mechanically lysing the cells or whether you are chemically lysing the cells, that also has an effect, or sometimes a hybrid approach can also have a better impact. And DNA purification could be in a silica column base, like how they have in QIAGEN or something like the Promega, which has already been discussed by people in the lab in the talks earlier, magnetic based approach. Everything to get your sample, and then you do quantification, qubit, nanodrop, and then you go ahead with the PCR. These are the machines and the kit that you could use. And essentially, if you take the Promega method, you add the sample and the lysis buffer, you put it in the heat bath, you do a centrifuge, you get a lysate, you put the lysate in the cartridge, like how they had mentioned earlier, and then you put it in the machine, and then the machine does the extraction for you. Once you get this DNA, genomic DNA out, like so, you put in your 16S primers, which are already there in your kit. It depends on what kit you're using, and Oxford Nanopore has their own 16S barcoding kit. They have been generous enough to give you barcode included primer sets, so you can deal with multiple samples. So the primers get incorporated. They have an adapter region to which the sequencing adapters can then be attached using their rapid chemistry, and then you can then proceed to loading. And this is just another easy schematic for the same. Any questions here? Because this is a very simplistic, brief overview anyway. This is amplicon. This is 16S amplicon, because you already know which region you want to focus in all the genomes in your...
You didn't know the region, then you would have chosen shotgun.
Absolutely. But then again, this is nanopore, so that shotgun does not really apply here. Yeah. Just a visual representation, that is the Flongle flow cell, which kind of fits in the palm of your hand. You then proceed first by doing a priming of the flow cell. They provide all these reagents with the kit. You then load the sample later on, and then it is already hooked up to the computer, and then you just hit start sequencing, and it will do the work for you. Dr. Gaurav has already explained these schematics. There's that pore, and then the motor protein will tether the strand to the pore, and then sequencing occurs, and then you proceed with bioinformatics. Just a very wonderful picture. I thought it's just visually very pleasing. Yes, you read the musical notes of nanopore sequencing. And then quickly concluding with why to do 16S long read sequencing with nanopore platform. It says by itself, you have the advantage of a longer sequence, and the real time data analysis has its own perks. If you ignore the amplicon sequencing, then the native DNA sequencing avoids all the amplicon bias, amplification bias. The portability is also very handsy, and it can save up other kinds of costs. And besides all of that, you also get better taxonomic classification if you do the same data analysis using Illumina. Now, if you remember that V1 to V9 region, this will be covered in more detail in depth by Arpit in the later talk. But in essence, what Illumina versus Nanopore could do is, let's say Illumina can tell you, okay, your sample has 5 species of, let's say, the genus Enterococcus, but a better taxonomic classification or resolution by Nanopore will tell you, okay, no, the 5 Enterococcus and Enterococcus faecalis, Enterococcus, Pseudomonas or Aeruginosa, and so on and so forth. So that is the added advantage of Nanopore long read sequencing as opposed to the Illumina V3, V4 gold standard method that is out there. Hence giving us a comprehensive microbiome profile of any sample that you wish to. That is pretty much it. This kind of information comes with collective efforts of everybody in the lab, current and past members. And yeah, the floor is open to questions, and please proceed to tea. Thank you. We need not discuss right now. We can always disperse. Thank you. Yeah, already fine.
Let's take a short tea break to refresh and recharge, and we'll reconvene at 11:20.